Good evening, everyone, and welcome to the Real Science Exchange, the pubcast where leading scientists and industry professionals meet over a few drinks to discuss the latest ideas and trends in animal nutrition. We're here at the ADSA scientific meetings in Ottawa, Canada this week, and we've been having conversations with students all week about the abstracts that they've submitted. Now, this is part two of two parts. So if you've missed part one, please go back and listen to that. My co-host for this session is going to be Dr. Pete Morrow. He's a tech service representative for Balchem. And my guest today is Dr. Heather White and Dr. Billy Brown. Heather, you really need no introduction, but if you wouldn't mind, give an introduction to our audience, and then also tell us a little bit about Billy. How'd you get to know him and some of the work that he's done for you?
Yeah, great. Thank you, first of all, for having us here and for your interest in the research. I'm Heather White. I am faculty at University of Wisconsin-Madison in the area of nutritional physiology, so I'm a professor there. Just hit my 10-year mark earlier this year, so that's good. Do teaching and research there and serve as the faculty director for the Dairy Innovation Hub. So as a part of my research program, always looking for great additions to the lab, grad students, postdocs. Billy joined the lab and bravely joined without ever having met any of us in person, thanks to COVID. So Billy and I met by Zoom, and we just had a lot of trust in the situation there. He may or may not tell you that was well-placed trust, but he came to Wisconsin and jumped right into a cow study. It was, in fact, the cow study that yielded the calves we'll be talking about today. So he spent about 1.5 years with us in Wisconsin and is now faculty at Kansas State. So let him tell you a little bit more about himself.
Yeah, go ahead, Bill.
I was really grateful that they weren't all holograms. They were real when I moved to Wisconsin, but no, I'm a faculty member at Kansas State University. I'm primarily teaching, 60% teaching, but do have a research appointment, so we're trying to get that off the ground, launching that now. And we'll be starting a calf starter grain study with my first grad student here in a couple of weeks, so we're excited about that.
All right, very well. Hey, would you mind giving us then an overview of the abstract that was presented here? Was it a presentation or a poster?
A poster.
It was a poster, yeah.
Okay.
So we're really excited about those results, but and we've had a number of abstracts that have resulted of that. But to kind of give you some cliff notes, we fed several varieties of choline, the traditional Reassure, and then a prototype of that, more concentrated prototype, to prepartum dairy cows for 3 weeks prepartum, and then we took the resulting calves from those cows and tracked them through a period of time. And specifically for this poster that we're talking about, we were looking at Holstein and Angus cross calves. And so we tracked their growth for a number of months, and then we fed them a finishing diet, and then we took them to slaughter and looked at carcass characteristics of those animals and how that changed their carcass overall. So really the first of its kind research in this area, and we're really excited to be able to be a part of it.
Okay. Just to kind of summarize, so these animals the mothers were fed the choline,
Yep.
prepartum, and then the calves then no
Yeah,
choline, or what was the?
They all had a standard diet. They were raised similarly, so all the treatments were really based upon the dam feeding while those calves were in utero.
So this,
So this
epigenetic
concept,
kind
this
of
epigenetic thing,
yeah.
this concept developmental programming, which is starting to gain more traction, and we really have the perfect environment with the dairy industry to really isolate these gestational effects of the things that we can do through nutrition or management. And really isolate those compared to other species, because we do raise those calves on a bottle in a very homogeneous way throughout the rest of their lifespan. So a great model that we can leverage.
I'm kind of curious, did you have a hypothesis ahead of time of what kind of results you would see, or were you just kind of going into it to see what you would see?
Well, one of the things that we did know was looking at some past literature from Florida, we had a good hunch that calves might have a little bit of a growth advantage. And we did see that in the first 10 months where there was some growth advantage. But other than that, we didn't really know what we were going to see. And so one of the big things that we did note, I'll go ahead and jump into some of those results if that's okay, is that at the time of harvest, at least, it didn't appear that there was any change or evidence of difference in the weight of those calves. or the ribeye area, but we were seeing some differences in what appears to be fat metabolism. So they had greater kidney, pelvic, and heart fat, but they also had greater marbling. And that's the part that really excites us, because there's very few ways to influence that marbling outside of genetics, or just feeding them a higher energy diet, right? We know that Wagyu and Angus are great at marbling, but nutritionally speaking, that's a little bit tougher nut to crack. And so we did see an increase there, and I think that had some pretty big implications long term for the beef and dairy industries when we talk about beef and dairy animals.
Heather, I'm just kind of curious if you've got a hypothesis as to what the mode of action might be.
Yeah, so we measured quite a few metabolites along the way. So Billy talked about the carcass quality and carcass characteristics. But we also took blood samples, and we found some differences or tendencies for differences in things like insulin and glucose. And so when we're thinking about that, marbling plays into that. And like Billy said, we think this is probably related to glucose and lipid metabolism. That doesn't surprise us if we think about what we've seen in the lactating dairy cow, pre and postpartum, and when we think about some of the cell culture work we've done to look at the mechanism of action. When we supplement cells with choline or in vivo, we tend to see changes and differences in glucose metabolism and lipid metabolism that support milk production through increased gluconeogenesis, increased oxidative capacity, increased VLDL export. So it wouldn't be surprising to think that that could be a hypothesis for mechanism here. We talked about epigenetics earlier. We were also really interested to see if methylation patterns had changed, because that's, with choline being a methyl donor, that's also something that we would suspect. We looked at global methylation patterns in blood samples, so not targeted, not single gene up or down regulation, just global methylation patterns. And we did observe an increase for that in the male offspring. We did not observe a difference in female offspring of either the Holstein or the beef cross. Now, at first, that's a little surprising, because we saw changes in growth across all of the offspring. But we have to remember, first of all, that there were a lot of metabolic changes as well, and metabolites that were changed. So there's probably more than one mechanism of action here. But also, we know that across species, rodent models, sheep, humans, that the male offsprings are more likely to have increased global methylation and be able to detect that. The females are much harder to detect the difference in methylation pattern, even if there's a difference in the growth outcome. So again, this is consistent with that across the board. So at this point, I think there's probably a little bit of both of those, both through methylation pattern changes, and we should definitely dive into those more in the future, and then maybe directly through changing glucose or lipid metabolism pathways.
Do you think you'd see similar results from feeding choline to finishing cattle, or is it just an epigenetic response?
Yeah, I mean, that's a little bit of a tough one to guess, right? Because we probably have the opportunity for a big impact through the fetal programming. Like Billy mentioned earlier, that's an opportunity for us to impact the animal when it is both going through a big growth phase in utero, but also setting kind of the stage for lifelong growth and metabolism. And so if we have an effect there with something, we don't always see the same effect, depending on what nutrient we're talking about, later by feeding it directly. But to my knowledge, we haven't fed, or at least in the published literature, maybe there's some stuff hiding we can dig out, but in the published literature, there's not cases where we fed choline in the finishing stage in controlled randomized studies. So we either need to go back and see if we've done that in the past, maybe in beef animals, and we can look at it again here in the future. So I think it's probably worth exploring.
Yeah, thinking about a future, so I guess there's a lot more things to understand. What does the next step in research look like?
Yeah, well, I think the world is our oyster at this point. These are exciting results, but they're, in a way, fairly preliminary results too. So I think, some of this is just maybe taking a different approach at it, maybe looking at a different stage or a different species, and trying to see if we can replicate this at least. And then I think diving into some specific tissues and understanding what's happening in those tissues metabolically, what's happening from DNA methylation standpoint, and trying to get a better picture and digging down deeper into those a little bit better. So it's one of those situations where there's so many different avenues you can start with. Where do you start to narrow it down a little bit?
Yeah.
So.
I think if I can add to that, one thing to point out is that this is a fairly small sample set. By the time we break it down, we had 4 maternal treatment groups. Each of those, so even though there was 24 cows in each treatment, which is powered for that, then we have offspring. Half of those are Holstein heifers, half are beef cross, half of those were female and half were male. So by the time we get to those final carcass characteristics, some of the treatment groups maybe had 12 animals in those groups, right? So first and foremost, we've got to recognize that this needs to be repeated. Just like Billy said, we need to make sure that this can be replicated across and that we observe the same thing. But I think that goes back to the original reason why we did this. So the beef cross animals following to slaughter was never a part of the original experimental design. In fact, we were only ever planning to follow all animals to weaning. But one thing for me is that it was really full circle. My master's is I had a meat science component and lipid metabolism component, spent a lot of time looking at both swine and beef carcasses. And we saw that out in the real world, people are breeding these beef cross animals, and there's a complete dearth in benchmarks, right? There's not those average metrics for people to look at and say, okay, these beef cross animals are growing as we would expect. I'm meeting the benchmarks. I'm not. I'm average. I'm below. I'm above. And people really needed that data. And so we had an opportunity to do that. And for me, it was kind of full circle to go back to something I had looked at years ago. With Billy joining the lab and some background in beef and the ability to add capacity to the team, we were able to look at that. So a big part of it was just to add baseline data, right? To get some data in the literature and to start building that database on what's there. The fact that we observed differences in slaughter from a prepartum maternal treatment was really rather exciting to be able to see significant differences from something we never fed to the calves themselves. So that was really a pleasant surprise because we really were just looking for some baseline data. So first and foremost, I think we've got to make sure that this is repeatable across and then dig into it and see what kind of nutritional power is there, right?
Yeah, very exciting. You know, choline's one of those compounds, the more we learn about it, the more we learn that we don't know a lot about it. So there's a lot left to know. I really appreciate you guys stopping by and spending some time with us today. So thanks a lot.
Thanks for
Thank
having
you.
us.
Guys.
Yeah, thanks.